All right. Hello, I am Christopher Groff, and I will be talking about healthy low-carb diets and time-restricted eating interventions and their effects on anthropometric values in the gut microbiome. So this is my paper below. We're specifically focusing on how these different diet interventions are going to help adults who are either overweight or obese. So first, let's talk about hypothesis and significance. So when looking towards the hypothesis or research question put forth in this article, along with the significance of the article, we must first ask why we should care about obesity and its relationship to diet and also microbiology. So obesity is a global health crisis linked to numerous metabolic diseases, and understanding effective dietary strategies for weight management and gut health is crucial in addressing the growing challenges associated with obesity. The question being asked in this research was whether a healthy low-carb diet, which will further be abbreviated as HLCD, and also time-restricted eating, which is a type of intermittent fasting, which will be abbreviated to TRE, alone or in combination, are able to enhance weight loss, body composition, or maybe positively alter the gut microbiome beyond the extent of a normal caloric restriction diet. So that would just be cutting some calories. So this study, as you'll hear, focuses on adults who are overweight or obese. And the significance of the study is that it provides evidence that dietary patterns like this HLCD and TRE can offer additional health benefits beyond the deficit diets, especially in the gut microbiome, potentially improving long-term weight management and health outcomes. Some necessary background understanding of obesity and dietary interventions is needed for this article, particularly low-carb diets and time-restricted eating. So obesity is a global health challenge that has been rapidly increasing in prevalence, particularly in China, where this study was conducted, where central obesity and associated cardiovascular risks are rising. So moderate caloric restriction has thus far been the standard recommendation for obesity treatment, and the relative benefits of specific dietary patterns beyond this have been pretty much unclear in research. So low-carb diets have been shown to be effective for short-term weight loss and improving cardiometabolic biomarkers, such as reducing triglycerides, increasing HDL, cholesterol. However, most studies focus primarily on the quantity of carbs, whereas this study is going to focus on the quality, hence the term healthy low-carb diets. So the impact of low-carb diets on the gut microbiome has also been mixed, as some studies actually show the potential for negative effects like reduced bacterial diversity, while others note some beneficial changes, mainly in functional microbes. So time-restricted eating, which in the study was performed as a 10-hour eating window during the day, has been shown to be effective for weight loss and also improves insulin sensitivity and reduces inflammation. So long-term sustainability and effects beyond caloric restriction of this diet have also been limited in research. And there are some animal studies of TRE diets that have suggested gut microbiome improvements in diversity and function, but so far human trials have been inconsistent. So in general, diet is a crucial factor influencing the gut microbiome, with high-quality diets rich in fiber and polyphenols generally promoting beneficial microbial profiles. The gut microbiome plays a significant role in metabolic health, with positive alterations linked to lower metabolic risks of obesity and other disorders. So this study is attempting to fill the gap in that knowledge, in the lack of well-controlled, especially isocaloric feeding trials that examine the effects of specific diets on weight management and gut health, rather than just simply cutting calories. So the 20-week follow-up in this study also intends to help with predicting the long-term sustainability of these diets, and the interventions, and also their effects on gut health and metabolic outcomes. The potential benefits of combining HLCD and TRE interventions were also largely unexplored, and whether metabolic and microbiome improvements would sustain after the cessation of these diets are also unknown. So that all goes into why the follow-up would be necessary in this case. The urgency and broader implications of this study, as touched on earlier, rely on the growing crisis that obesity represents, as it's estimated now that 1 billion people in the world are obese, and this is driving up rates of type 2 diabetes, cardiovascular disease, other metabolic disorders. So this study could lead to emphasizing of high-quality carbohydrates and plant-based fats and low-carb diets as more important than just eating in a calorie deficit to optimize fat loss while also preserving lean mass. So gut microbiome changes like reduced fecal branch chain amino acids or BCAAs, and increased unsaturated fatty acids also could act as biomarkers for future personalized nutrition approaches to predict dietary responsiveness to interventions. So this is something I'll get to later. And then gut microbiome alterations could also offer targets for possibly new therapeutic interventions. And this could help address the critical challenge of weight regain after the interventions. This is why we're looking at the follow-up in this case. So by linking diet quality, timing, and the gut microbiota, this research could help push us towards precision nutrition and also give us actionable strategies to combat obesity-related conditions. So now for the methodology of this experiment. So this is shown on the flowchart on the right, as it was conducted first with pre-screening, and then there was 4 visits throughout the trial with the doctors. So this was a 12-week randomized controlled feeding trial, and it utilized 96 overweight or obese adults is how they're classified. So the trial also included a 28-week post-intervention follow-up, which was going to help look into sustainability of the diets. So participants were randomly assigned to 4 groups. So it's either the HLCD alone, the TRE alone, or a combination of both, or a controlled diet. So all were isocaloric, with meals provided on 5 weekdays, adding up to 1,600 calories and 1,300 calories a day for men and women, respectively. The HCLD or HLCD emphasized high quality carb sources, and other calories were mainly from unsaturated or healthy fats, and also plant-based proteins. And then the TRE required eating within a 10-hour window. So then some information about the subjects. The mean age was 36.3 years old, and the mean initial BMI was 26.9. So each participant was also assigned a researcher in this case that ensured a high adherence to the dietary patterns. And then the flowchart also notes that a limited number of participants had to drop the trial for multiple reasons. So now, kind of with the methodology, we're going to have the microbiome techniques used in this paper. So we begin with the sample collection, which was done through fecal samples taken at the baseline, the week 12, which was after the feeding intervention was finished, and then the week 40, so that was after the 28 post-intervention window was closed. So that was the follow-up. And then whole metagenome shotgun sequencing was performed by BGI, which is a Chinese genomics organization. So they were using their DND-seq technology, which sequences by synthesis with DNA nanoball technology. So quality control processing was also used, as software such as NeatData, Trimmatic, and Bowtie2 were used to filter human and rDNA reads out. So this technology used the DNB-seq was next-gen sequencing, most similar to kind of Illumina, as we learned in class, in terms of like short reads, high throughput, but BGI avoids the PCR bias by using these nanoball technologies, and they also have comparable accuracy to Illumina, but it comes at a much lower cost. So targeted metabolomics was also performed using ultra-performance liquid chromatography, which helped identify fecal metabolites in this study. And then the statistical approach for this study, so it employed multiple different methods to try to address some confounding variables, such as using mixed effects linear models, which accounted for repeated measurements, and then also individual variability within those measurements. And then they also used age, gender, and then also the baseline values as covariates for all the analysis. And then microbial community composition change was also performed, as well as identification of and the abundance of different microbial species were noted between the different time points. So the strengths of the methodology in this case include the controlled feeding nature and the isocaloric meals that ensure there's a consistency in the caloric intake, and then the use of like multi-omic integration to combine microbiome, metabolome, and then clinical data together was definitely a strength. And then as well as the 28-week post-intervention follow-up for sustainability. And then finally, the factorial design, isolated effects of the HLCD and the TRE diets. So this allowed for individual comparison. So you actually see which factor is causing the changes, the statistical significance. And then some limitations included So there was a limited statistical power for detecting between group differences in the microbiome because the relatively small sample size of 96. So the metabolic healthy nature of the overweight people selected in this study also could bias results because this would kind of bias it away from people who are at more risk populations, so possibly people with metabolic disorders were not selected for this study. And then also the short intervention period, so it was only 12 weeks, so it may not have captured all long-term adaptations in the microbiome. And then the limited caloric nature of the diets also caused some nutritional deficits, and this was noted by the authors. So some things such as fiber intakes were slightly below what would be recommended. So the first results I'm going to focus on are those that are more the anthropometric indicators. So these are like body composition measurements. These are shown on the right. So first thing to note is that weight loss was noted across all 4 groups, ranging from 2.57 kilograms in the control group to the highest being 4.11 kilograms lost in the combined both intervention group. So while all groups achieved significant weight loss due to the caloric restriction, the HLCD caused greater fat mass loss by almost a kilogram greater than non-HLCD groups. So TRE, while not significantly affecting fat mass, did actually lead to lean mass loss. So the implication of this being that emphasizing high quality carbs like whole grains and vegetables, and then having the other calories come from unsaturated fats and plant proteins in HLCD, this selectively targets adipose tissue. And then, therefore, HLCD is the best option in the study for sustainable fat reduction and general metabolic health markers. And TRE would actually require very careful consideration because if it is implemented, it could possibly lead to muscle loss. So, as we move to a more microbiome-based results, I'm going to walk through each panel on this figure more slowly. So this is going to generally demonstrate how the different dietary interventions, so the HLCD and TRE, were distinct in how they reshaped the gut microbiome during the feeding trial. So graphs A and B show the general composition changes as well as the significant beta diversity shifts that occurred in the groups, but that did not occur in the control groups. So this was only in the HLCD and TRE groups. So this indicates that the dietary patterns, not just the caloric reduction, was what drove the microbial restructuring. So HLCD also significantly reduced the FB ratio, which is a marker strongly associated with obesity. And then TRE also showed more beneficial probiotic species. So this included species that are known to produce short-chain fatty acids. So then taxonomic analysis shown in the graphs in C and D also indicated 9 species level alterations in the microbial bacteria for HLCD, and then there are 11 shown for TRE. So most of these, like some of these of note, would be there was a decrease for one species in HLCD, which was associated with obesity in some Western studies. And then there was also an increasing in an anti-inflammatory bacteria that was in TRE. So then for charts F and D, this is also a focus. So these show the actual functional pathway alterations. So HLCD was primarily affected in pathways involving purine metabolism and fatty acid biosynthesis, while also affecting carbohydrate degradation. And then TRE influenced amino acid biosynthesis and carb degradation as well, but also energy metabolism pathways. So the meaning of these changes are that HLCD impacts the pathways involved in fatty acid and purine degradation, which this would kind of explain its fat mass reduction, while TRE that affects amino acid biosynthesis, you could then relate this to maybe possibly why it showed lean mass loss outcomes. So now, as we look at the metabolome rather than the microbiome, we're going to look at how these alterations may relate to the actual body composition measurements. So results regarding the metabolome are shown in this. kind of dense figure, but again, I'll go through each panel. So in panel A, we're going to see that the changes in some representative metabolites across all 4 intervention groups were shown. So things to note from this panel include HLCD shifts that showed the most pronounced effects. So again, branched-chain amino acids, or BCAAs, they decreased significantly in the HLCD group. So also unsaturated fatty acids did increase significantly in HLCD, and then TRE had much more modest effects, and this led to the rest of the figure generally focusing just on the healthy low-carb diet group. So panel B is looking into HLCD versus non-HLCD metabolite differences. So this is just showing only the significant groups. So again, shown are decreased BCAAs, but also decreased indoles, which this is related to inflammation, and then reduction in phthalic acid, which is linked to many metabolic disorders. And then again, there are more unsaturated fatty acids, and this is all reflected in the more healthy low-carb diet. So these differences point to HLCD's potential mechanisms for improving metabolic health beyond just weight loss alone. So panel C is going to show metabolite correlations with clinical parameters. So this is going to visualize how BCAAs, phthalic acid, and other residues are strongly correlated with decreases in body weight, BMI, and fat mass. So these kind of correlations suggest that specific metabolites might serve as biomarkers or even mediators of weight loss efficacy. So I'll go into this later into the discussion, but as I go to panels D and E, we're going to get into the multiomics kind of part where we're going to integrate the taxonomy, the pathways, the metabolics, and then we're going to visualize all these connections together. So the correlation network shows kind of the relationships between the microbiome and the clinical measures in the study. So one such example is that BCAA is inversely associated with a particular strain, with a particular species found in the gut. That abundance was positively correlated with BMI, fat mass, and serum triglycerides. So the significance of metabolomics findings are that they may provide these mechanistic insights into how these diets actually do affect weight and metabolism. So a couple of key points that I found from this figure were that the HLCD's BCAA reduction likely explains the fat mass loss, as BCAAs are associated with obesity and insulin resistance. And then the increased unsaturated fatty acids also likely provide cardiometabolic benefits that go beyond just weight loss. And then also TRE and having more of a modest alterations to metabolites, again, aligns with the lower effects that this diet had on fat loss. So these metabolomic changes help to explain the observed clinical benefits and also provide as possibly potential targets for future precision nutrition approaches that I'll talk about in my conclusion and key takeaways. So the final portion of the results is going to be talking about the post-intervention 28-week follow-up analysis for specifically the HLCD. So this could be kind of talking about the more long-term effects and how this is sustained. So this figure shows that certain effects of the HLCD persisted into the 28-week follow-up intervention. So this kind of shows implications for long-term dietary interventions. So panel A and B show a sustained reduction in fat mass and fat percentage. So these panels show that changes in fat mass and fat percentage from baseline to post-intervention, like the follow-up window. So this is over 40 weeks. While the non-HLCD groups gained fat over this same 40-week period, the HLCD groups maintained that reduced fat mass. So this resulted in a significant difference between the groups. So then similarly, this is reported for fat percentage, where the HLCD groups were able to keep off the fat percentage, while the non-groups showed an increase and gain it back after the initial 12 weeks. So this kind of a remarkable finding, because it demonstrates that HLCD's effects on the body composition, like they kind of persist even 20 weeks after the intervention cessation. So this kind of challenges the common like weight loss regain cycle that's seen with most diets. So the sustained fat loss suggests that HLCD may induce possibly like metabolic adaptations that extend much beyond the intervention period. So panel C shows persistence and alterations to the gut microbial functions. So this is going to show the top 15 microbial pathways that remain significantly altered into this 28-week post-intervention window. So there were pathways suggesting lasting effects on nitrogen metabolism, lipid processing, and these kind of provide explanation for the diet's lasting effects on body composition, as these can influence the host energy metabolism and then also inflammatory processes. So panel D is going to show the alterations in metabolites themselves that remain the same, noting that phthalic acid decrease and hydroxyphenylacetic acid increase are kind of like the two biggest things from this panel, and this is going to work to promote beneficial gut bacteria, and it's also going to stop some metabolic dysfunction. So this is shown as a persistent change. So these suggest that HLCD induces lasting alterations in the microbial host co-metabolism. So this potentially contributes to improved metabolic health over a sustained period of time just from this small intervention. So panel E is finally going to show the between group differences in the metabolites, and it's also going to reinforce the sustained reduction of the phthalic acid, showing that the intervention provides possibly longer term like detoxification benefits. So these findings collectively demonstrates that this diet induces persistent changes in the gut microbiome function and metabolite production, and that this correlates with sustained reduction in fat mass and also provides insights onto how dietary quality, not just the quantity, can have lasting effects on the metabolism beyond just the original intervention period. So what does this all mean? So again, this study kind of demonstrates that a healthy low-carb diet, or HLCD, and also the time-restricted eating, the TRE, offer additional benefits for weight management beyond just a calorie-restricted diet. So with HLCD primarily reducing fat mass, and then TRE causing some lean mass loss, were pretty significant findings. So the sustained effects of HLCD on fat mass reduction also highlights potential for a long-term metabolic health benefit and shift in the body. So these benefits are at least partially explained by the gut microbiome and metabolic pathways that were discovered in this paper to be linked to this diet. So these findings have significant implications for obesity management and suggest that diet quality and timing are both crucial for sustainable weight loss and metabolic health. So this study underscores the importance of personalized nutrition as well, because these strategies consider both the quality of the macronutrients and then also the eating patterns for each individual person. So some strengths that I found again in the study for extrapolation include the controlled feeding design. So this ensures compliance, reduced bias from like self-reporting. The multi-omics approach is also very good because it integrates many different disciplines. So it integrates ideas from the microbiome, the metabolome, and then also clinical data to provide a more comprehensive insight and look into the diet microbiome interactions. And then also the long-term follow-up. So again, this just assesses like more sustainability ideas and the effects post-intervention of these diets. And then limitations. So I kind of poked at these earlier, but the sample size. So again, there is limited power, especially for the microbiome analysis. And then this is just due to a smaller sample size. And then population bias. So again, the participants were all metabolically healthy. So this kind of limits the ability to generalize to possibly like a more high risk population. So maybe those with metabolic conditions. And then the short intervention period. So you're not going to capture every single adaptation in just 12 weeks to the gut microbiome. And then for some possible ideas for future directions, I think that increasing the sample size obviously would enhance the statistical power. And then a longer intervention period, of course, would help you see maybe different long-term microbiome adaptations, and then possibly including high-risk populations. So if you include those with metabolic disorders, maybe you're going to increase how generalizable it is. And then I think a mechanistic study would be interesting. So if you investigate specific mechanisms by which the HLCD and TRE diets influence metabolic health, that could be interesting. I also think maybe investigating protein supplementation or resistance training to help mitigate TRE, and then see if that would then be good as a recommendation for a dietary intervention. And then I also think exploring microbial transplantation to determine like the actual causality of the microbiome changes and metabolic outcomes would be interesting. And finally, I think that additional studies could be used to develop personalized approaches. So using baseline microbiome profiles, studies like these, we could help predict like dietary responsiveness, and then also examine whether a specific bacterial species, maybe even some identified in this, could be used as a therapeutic target. So my kind of personal takeaway was that the study fundamentally shifts our understanding of weight management by like demonstrating how fat loss depends not just on how much we eat, but also what we eat, when we eat. So the persistence of microbiome changes with HLCD suggests that diet quality may reprogram the gut host metabolism for lasting benefits, while TRE's impact on muscle reinforces that weight loss composition matters more than just the kind of like just the numbers on the scale. So I also put these 5 questions for group discussion. So kind of just to quickly go through them. My first one's kind of about the mechanisms for these diets and why I say in this one there are particularly more benefits for HLCD as opposed to TRE. And then also pointed out how what about like the reduction of BCAAs? So this was pointed to multiple times as associated with HLCD, and how this may be used as a method for health interventions, and how this could be like sustainable over a long period of time. Also was wondering what thoughts would be about whether TRE is a good recommendation for people with obesity, because we saw in the long term, what if it leads to lean like mass loss, and is this an actual like a potent risk to these populations? And then finally, how should future studies address the limitations of this trial, particularly like sample size, population diversity? So is it important to include those with metabolic conditions, or should it still mainly now be focused on those who, although are overweight, do not have any of these conditions? And then finally, I was wondering if other diets would compare similarly, especially in how they induce microbiome changes. So, what about diets such as the Mediterranean diet? How would that introduce similar or different lasting microbial benefits in the body? And then finally, again, to kind of conclude of like why we should care. So again, we're in an era where obesity affects over 1 billion people globally. So understanding the sustainability of dietary approaches is very crucial. So we should care about this study because first it shows how diet quality and HLCD specifically can target fat mass rather than just general weight loss. That would be like happening in a traditional just caloric deficit diet. So this study was also going to show how eating timing can affect us metabolically, as TREs like lean mass loss highlights how fasting protocols really need to be carefully considered if they're going to be implemented. This study also kind of identifies that specific microbial changes may explain these metabolic benefits beyond calorie restriction. So it also kind of highlights the potential for microbial-based personalized nutrition approaches in the future. So finally, the persistence of certain microbial changes that were shown into that 28-week post-intervention window kind of suggests that the gut microbiome may almost like remember these dietary patterns, and this could potentially explain the HLCD's lasting benefits and also offer a way for us to give long-lasting results to people who try to go on these diets. So this research suggests that we need to move beyond the simplistic kind of like calories in, calories out approach to a more sophisticated approach that considers diet quality, timing, and then also like individual gut microbiome responses. So the fact that HLCD's emphasis on high quality carbohydrates and then also plant-based fats, how this led to a sustained fat loss, offers a promising direction for developing evidence-based dietary recommendations. And then as a closing remark, as we confront the global obesity epidemic, This study kind of like underscores that sustainable weight management requires attention to both the composition and the timing of our diets, and mainly like the idea that the gut microbiome serves as a potential therapeutic target, especially for the future. So by focusing on HLCD's fat-specific sustained effects, we may be able to develop more personalized and microbiome-aware dietary strategies that can address obesity at its roots. And here are my references.